Hello, everyone, and thank you for attending today's webinar, Are Real-Time Gene Therapy Test Results Possible? Presented by Catalent. I'm Michael Shoshua, and I'll be moderating this webinar. Our speakers today are Bhargavi Kundragunta, Director of Internal R&D and Product Development at Catalent Cell and Gene Therapy, and Sean J. Hart, Chief Scientific Officer at LumaCyte. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Now let's begin. Sean, please go ahead.
Thank you very much, Michael. It's a pleasure to be here. Happy to have everyone on the webinar with us. I'm going to be introducing LumaCyte and laser force cytology in our Radiance instrument, and Bhargavi will be talking about their use of the instrument for a number of gene therapy applications. So let me jump right in. So laser force cytology is a very disruptive single-cell analytics platform, and I'm going to describe the scientific basis for you and run through some key applications and some data to set this up for Bhargavi to share with you her applications. So laser force cytology is single-cell analysis based upon measuring intrinsic biophysical and biochemical properties of single cells. And so what you're looking at in this video are vero cells infected with the Zika virus traveling through the microfluidic channel in the Radiance instrument. And so they're being driven in the system by fluidic flow from right to left, and they encounter an optical force from a laser beam on the left-hand side of the channel. And so as the cells move through the channel, they experience this optical force. And optical force occurs because photons have relativistic mass. They're moving at the speed of light, so they have a tiny relativistic mass that they can impart on cells and generate a physical force. And so one photon has a tiny relativistic mass and a tiny force, but when you have 10 to the 20 photons at a wavelength that's transparent and doesn't damage the cells, you can generate hundreds of piconewtons of force to slow cells down or stop them in a fluid flow. Now, in Radiance and laser force cytology, the cells aren't stopped, they're merely slowed. And the degree to which they slow down, the velocity change, is proportional to the optical force. And so the optical force is related to changes in intrinsic biophysical and biochemical properties of these cells. And we're measuring not just velocity, we're measuring dozens of parameters related to where the cells slow down, where they come into the beam, where they leave, and how they rotate or how they orient themselves in the channel, shape parameters, and a number of other ones that allows us to build really sophisticated multivariate models that are label-free. And so there's a number of applications that we've been developing over the last number of years in vaccines, R&D, and manufacturing, but more recently in cell and gene therapy. Now, the instrument itself is making these single-cell measurements based upon optical and hydrodynamic force measurements, right? And this allows us to access new cellular information. We're measuring, a couple dozen parameters, including cell velocity proportional to the optical force, the shape, the deformability, and this allows us to use very robust multivariate data analytics, things like machine learning, to make, incredibly robust models to predict, processes. And the system itself allows for automated data collection. It's 21 CFR Part 11 compliant. It uses a 96 well plate format. It's flexible in terms of the cell input, right? You can use suspension cells or adherent, just lifting them off with trypsin or trip-LE or something like that to get them off the surface so they're suspended just like you would in flow. It's a benchtop form factor, as you can see here. And the types of things that we're measuring are unique biophysical and biochemical cellular responses, right? Changes in organelles, changes in the nucleus, detecting the presence of viral inclusion bodies, for example, which is very important for, viral infectivity, obviously. As virons are produced in a cell, they increase in number, and that changes the refractive index due to the high protein content building up within the cell. And we're exquisitely sensitive to that. So this allows us to detect this much sooner, than other technologies, and certainly sooner than the gold standard plaque and TCID50, for example. So changes in nuclear cytoplasmic ratios give rise to optical force differences, as do cytoskeletal changes. Cells aren't damaged, and they are viable for growth and further study, in the system. So that's an important point, to be able to collect those cells as well. A couple of examples are shown here. The one on top on the right is a red blood cell moving through the system. As it moves from left to right, it encounters the focal point of the laser beam, where it is retained in the system or slowed down. And in that process, it stretches because the fluidic drag forces are stretching the membrane while the optical force is holding it back. And so a healthy red blood cell will stretch roughly 3 times its nominal diameter, whereas an unhealthy red blood cell is one that's suffering from sickle cell anemia or malaria infection or something like that, or even an older red blood cell, right, which is less flexible, won't stretch to the same degree. And so we can identify changes in red blood cells due to disease states or condition, using laser force cytology. So the example beneath that is a cancer cell. This is a lung cancer cell line. And in this cancer cell line, you can see there's a large nucleus with a lot of aberrant DNA within it. As it moves through the system, you notice the immediate difference that it's not traveling in a completely linear path. It actually bumps up in the beam, and this is a vertical shift that we can detect with the system due to the rotation of the nucleus rotation of the nucleus towards the back of the cell as it moves through the channel, and you can see that by the time it reaches the focal point of the laser on the right-hand side of these images, you can see it actually poking out the backside, and this is approximately a 10% stretch of the cell. So it's probing, the biomechanical properties by pinching it between the fluidic drag force and the optical force. So this is just another example of the types of measurements you can make using Radiance. So I'll take a moment, because it's a label-free technology, I'd just like to talk about some of the benefits of using a label-free system. And I'll focus on, antibody-based systems that potentially have problems. Obviously, antibodies are incredibly valuable and useful in bioscience, but they're not without issues. For example, when you use an antibody and you attach a fluorophore and it binds to an antigen on a cell surface, it's not just there so that we can detect that cell, although it's very useful for that. But when that happens, cytokine cascades can occur, maturation, differentiation, all kinds of biological changes can occur in those cells. By using a label-free technology, you can protect the innate cellular response by using a label-free approach. As many of you know, nonspecific binding can cause false positives, and maybe more importantly, because the antigens aren't always uniformly expressed, the label may not be able to detect that, leading to a false negative. So a label-free approach reduces errors in assays through making unbiased cellular measurements. Additionally, antibody use requires prior knowledge. You have to have, you have to have developed the antibody in advance for a particular antigen, making new cellular discoveries impossible or difficult. By using Radiance and laser force cytology, you can find new cells, new phenotypes, and new cellular biomarkers by using this sort of agnostic approach, right? That's open to all types of changes within a cell, not just the presence of a particular antigen. And finally, label use is error-prone, time-consuming, and expensive, as many of you know full well. And by using a label-free system, you can deliver higher quality results with less time and cost. So we get the question quite often, how are we different than flow cytometry? And we use flow, we use lasers. It seems quite similar, but they are fundamentally different technologies. And so an experiment we did several years ago highlights that very, very elegantly. And this is looking at mouse raw macrophages that were exposed to 5-micron silica beads. And in this experiment, we incubated them with the silica beads and looked at how many were getting engulfed. And we thought initially that it would be between 1 and 3. And it turns out that some macrophages are much more capable than others, and it really highlights the ability of the system, but also the heterogeneity and genetic diversity of the macrophages and their ability to phagocytose. So when we looked at this under flow cytometry with side and forward scatter, we saw that the two populations, the control macrophages and those exposed to the silica beads, were identical, and there was no detection possible at all. By contrast, using Radiance, we see almost baseline separation between these two populations shown on the right. And this is because it really is a fundamentally different technology, right? You're not just looking at scatter, you're integrating optical forces over all of the contents of the cell, in this case, the silica beads that have been phagocytosed. So this is a very elegant example, and we did publish a paper in 2017 looking at not only this, but looking at phagocytosis of bacteria and other applications, highlighting the sensitivity of the technique. So I'll take a moment and talk about the broad application base for Radiance and laser force cytology, focusing on R&D and biomanufacturing. In vaccine production, obviously being able to measure viral infectivity quickly opens a lot of new avenues and possibilities. We've done a lot of work in viral neutralization assays on the clinical side for understanding vaccine efficacy. In cell and gene therapy, in addition to viral infectivity, right, we've used this for monitoring transfection. Bhargavi will show you a very excellent example of that in her section. We've done work with T-cell activation, potency assays, and more recently looking at iPSCs and monitoring differentiation. And this is where the deformability really comes into play and really shines because, excuse me, as cells are differentiating, that changes significantly and really helps elucidate these different pathways. In terms of biomanufacturing optimization, right, it's useful from the very beginning with cell bank characterization, understanding the number of passages and having a quantitative tool to characterize that. Rapid process optimization and scale up, looking at infectivity and understanding that optimization, being able to do that much faster than before. Being able to analyze in-process samples. If you have cells that are infected and you're in a bioreactor, for example, or some other process, you can look at not only cell health before they're infected, but you can get real-time infectivity throughout the process. And finally, adventitious agent testing is a relatively new area for us. We've had significant interest and adoption in industry, and we're very excited about that application. So the technology is well proven. It's been studied over the last 20 years. There are over 30 publications on the underlying technology. Just to highlight a couple, we published an article in the journal Vaccine using Radiance as a TCID50 replacement. We had another article published using it for oncolytic virus in Science Translational Medicine. In 2019, we were awarded the Vaccine Industry Excellence Award at the World Vaccine Congress for the best production process development technology. We're very proud of being even nominated for this, but even happier, obviously, to have been given the award, considering our competition consisted of multi-billion-dollar corporations, GE Life Sciences at the time, Sartorius Steadum, 3M, and others. So we were very happy to be selected by our peers and the vaccine expert committee that made this decision. Also in 2019, we were selected by Frost & Sullivan for the Global Single Cell Analysis New Product Innovation Award. Again, a very prestigious award to get from this company. And I will highlight now a couple of projects we have with NIMBLE. For those who are not familiar, NIMBLE is the National Institute for Innovation in Manufacturing Biopharmaceuticals. It's a public-private consortium where large pharma works with companies and government, with NIST and FDA to advance biomanufacturing. So we've had a couple of projects that have been awarded and completed already. The first was with Genentech and Carnegie Mellon for adventitious agent testing. The second was was a laser force cytology training course with Celgene and BMS and RPI. And then the two ongoing projects we have are for CAR-T immunotherapy, trying to understand changes within the donor cells, the T cells, and variations within those and how that affects the manufacturing process and ultimate success. And we're looking to find biomarkers to better predict that process and prescribe what should be done. This is with Celgene, BMS, excuse me, and MIT. And finally, we have a project that just kicked off this past October with Merck and CMU, looking at automating bioreactor monitoring. So developing a system to remove cells from microcarriers and automatically analyze them in Radiance is a very exciting one that we're just getting started on. So I've talked about multivariate data capabilities. Having multivariate data on a single cell basis gives us new information and new ways to look at cells and look at their behavior. And it opens up the world for quantitative machine learning, and we're doing this in a number of industrial applications for vaccine production already. And the ability to have prescriptive analytics, right, that will guide process optimization and bioproduction outcomes. Very similar to what I talked about for, the CAR-T project and predicting success of an immunotherapy. And if we look at this example here, there's a video showing calibration cells in the system, and we see a significant overlap. But when we rotate into the velocity dimension, we can see differences in optical force between all of these particles. We can not only see that difference, but we can see the spread within those cells. And this, for a complex bioproduction process, being able to look at data from different dimensions and then use that in a multivariate or machine learning with machine learning tools is incredibly powerful. So when looking at reproducibility as an incredibly important part of an analytical instrument, we've shown that the Radiance instrument is incredibly stable. We looked at vero cells and vesicular stomatitis infected vero cells over 90 days post-infection, and we found that the average velocity had a variance of less than 1%, which is incredibly stable. And what we tell our customers is that if the instrument is operating within specification, if you see differences between your control and your infected or treated samples, that is a biological change. It's not due to instrument variation. These are negligible compared to the biological changes we typically see. So having multivariate data is fantastic, but it's important to have a robust tool to be able to efficiently process that data. And so we have spent the last year developing Reporter, which, as the name would imply, is an automated report generation, data analysis and report generation tool that we've developed for processing data from Radiance. And so the Radiance Illuminate software that runs the autosampler and the data collection is 21 CFR Part 11 compliant, and that is fully integrated into the Reporter workflow, which is also 21 CFR Part 11 compliant. And so the Reporter platform allows you to define the method when you're setting up your well plate, and the data at completion is automatically uploaded to the cloud, completely hands-off, and processed according to the method, and a report is emailed to you with your titers or your results. And so if we take a look at a couple screenshots, you can see once you log in, you have the ability to make a new experiment, explore a method, right? And you can develop your calibration curves by looking, at data when you're first developing a method. You can look at bar graphs or scatter plots, but this allows you to develop the method, develop your calibration curve, save it, and then use it automatically, then forward. So I'll switch gears now and go through a few viral infectivity applications, that will set the stage for Bhargavi to talk about the work that they've been doing with Radiance. So as an overview, we've used this for live virus vaccines, viral vectors, oncolytic, vaccine production, oncolytic virus, and viral neutralization. Below is a list of, cells and viruses used in the system. We've done a lot of work with baculovirus as well. This is, again, a partial list due to client confidentiality. It's probably double that number of cell lines and viruses. So we have a broad base of experience to draw from, when we tackle new applications and new customer uses. So comparing the traditional TCID50 assay with a Radiance assay, if you start with a virus that's been produced and purified, To titer that, you would infect cells with a number of serial dilutions, typically 12 and 8 replicates for TCID50. Then you would monitor that over 3 to 15 days, depending on the virus and the cell line. Then you'd be checking and scoring wells. Right? This is a laborious, error-prone, subjective process where humans are making subjective judgments about what is CP and what is not. By contrast, with Radiance, you infect far fewer dilutions because the linear dynamic range of the instrument is much larger, than plaque or TCID50. The incubation time is 3 to 10 times shorter, right? Typically between half to 3 days. There's no monitoring and a simple 5-minute analysis in Radiance. And to give you an example, right, in terms of getting a faster titer for adenovirus, we detect this in as little as 1 day versus 10 for your standard plaque assay. So this correlates with plaque assay TCID50, as I'm going to show you in the next couple slides. You can analyze cells that perform poorly in traditional assays, which opens up new capabilities that you may not have had before, and it delivers better precision with lower errors, removing assay subjectivity. So an introductory example is shown here for vesicular stomatitis virus infectivity in vero cells. So if we infect at 0 hours and we start looking at 8 hours with Radiance, looking at the optical force index, which is a size normalized parameter that we measure, we see there's really not much difference between the two. But by 12 hours, right, the infected sample in red starts to shift to higher optical force index, and by 16 hours, there's a very significant difference, and there's a large portion above an optical force index of 40 that are completely separate from the uninfected control, and we can, just like in flow cytometry, gate on this and be able to use that as a metric to correlate with the infectivity. So taking a look at the same data a little more closely, we did a Radiance response metric, right, which is how the instrument responds with one or more parameters. In this case, it was 2 velocity and velocity standard deviation versus the hours post-infection. And we found a peak at around 16 hours with a broad range of MOIs responding well. And to give you an idea, this again was a 72-hour process, for this particular work that we published using TCID50, and we're able to accomplish this in 16 hours. Now, if we look at the curve shown here of the Radiance response metric versus your standard TCID50, this calibration curve, the first thing you'll notice is that there's a good fit, but more importantly, there are 4 logs of usable instrument response, giving the system a great ability to look at a wide range of samples with a limited number of dilutions. And so if you measure the Radiance response metric, and you can use the equation of this curve to then calculate the TCID50. And this is exactly what Reporter does, completely automated to generate a report of your TCID50 results. So taking this a little bit further with the same system, this is validation work that's ongoing at LumaCyte. And we are looking at the TCID50 per cell versus the Radiance response metric. You see the calibration data in green with a good correlation between them, and there's 3, known and unknown samples, sample 1, 2, and 3, with 2 dilutions each. And if we look at the average predicted titer, this compares very well with the stock titer. The log 10 error difference is 0.06. As many of you, I'm sure, know, that 0.3 to 0.5 log 10 error is standard, normal for TCID50 or plaque assay. So this is an exceptional result. If we look at, in terms of accuracy, in terms of the standard deviation, we have a 22% variance there, which is also excellent in terms of precision. So I'll talk now about absolute or direct titer. If you don't want to develop a calibration curve or you don't want to run your standard assays, which is understandable, we've developed a method to do this, using what's called the absolute titer calculation. And so we use Poisson statistics, and we use a tool that we've developed and patented to look at a sample. And so what's required is a larger number of dilutions, and also a sample that is reasonably expected to show, complete infection of that sample. And so that's shown here. If we look at the volume of virus added from 20 to 2, we see there's this 100% infected region shown there. And so this, calibrates the system to what is 100% effective. And then we can use the dilutions below that to calculate a TCID50. And so that's shown on the right-hand side, where the predicted titer is shown over a 2-month period. And the average log 10 difference between the predicted and known titers was a little less than 0.1, which again is exceptional. If you look at the actual and predicted titers, these are phenomenal results and represent game-changing capabilities, for gene therapy and viral infectivity. Okay, I'll switch gears here and talk about process analytics and production. So with Radiance, you can take a sample directly from an ongoing bioprocess that is a cellular-based process. So you can take the cell sample either automated, as I discussed with the Merck project, that we're currently involved with, or you can take it, with a syringe and load it into a well plate and run it in Radiance, and you can have an answer in 5 minutes. So this is the only real-time viral PAT in the world, right? This at-line bioreactor sampling will give you a titer within minutes. And so depending on the cell line and virus, this is 800 to 3,000 times faster than a traditional method. Again, game-changing in terms of capabilities for process optimization and process monitoring. Okay, so I will now just summarize some of the uses in gene therapy applications. Radiance has been used and can be used for transfection efficiency monitoring, right, for media selection and optimization, looking at transfection reagents and testing the various transfection reagents that you like to use, plasmid testing. Obviously, I've shown examples of viral infectivity, viral vector infectivity monitoring. In terms of process development, looking at viral vectors and their efficiency, and then the cell lines in that development. These can all be done using Radiance for gene therapy applications. So Radiance is detecting subtle changes in cells, that in many cases are not detectable by other methods, including non-CPE producing viruses. And Bhargavi will show you an example of AAV transfection monitoring where that is the case. So in terms of a summary, Radiance transforms the status quo. It accelerates development with faster titer determinations, right? It's high quality result, very precise measurements. They're non-subjective and accurate. For discovery, it's a unique, sensitive cell-based assay that can give you, help you find new cell phenotypes, look at single cell responses without antibody labels. It's not virus or cell specific. And I think most importantly, for in process, you have answers in minutes, versus days or weeks. So this is a really important point. And so with that, I'd like to hand it over to Bhargavi and let her talk to you about their experience and their applications, for cell and gene therapy.
Thank you for your introduction, Sean. Now I'd like to talk to you about our experience at Catalent Cell and Gene Therapy with evaluating the LumaCyte technology as a possible PAT tool. To bring this to context, there is an increasing need for faster in-process analytics in the gene therapy as well as other advanced therapeutic fields, especially with the increasing number of both preclinical and clinical candidates in various phases of development, as shown on the slide. There is a need for faster process development while still gathering in-depth process understanding for enabling rapid tech transfers and ensuring clinical production. At this time, we lack real-time assays that could be indicative of viral titer similar to a plaque or a TCID50 assay. For example, the baculovirus determination in SF9 cells is currently done using a plaque assay, which takes 7 to 10 days, and the adenovirus titer determination in HEK293 cells uses TCID50 in 96 well plates, and that takes 3 to 10 days. And currently, the AAV transfection efficiency measurement in HEK293 is only possible in the R&D setting with GFP labeling, and this is done using a flow cytometer. And currently, there is no assay available for real-time transfection efficiency measurement using a GOI plasmid during clinical process development. And based on these current assays, these are the areas of optimization that we've identified. So we identified that there is a need for near real-time process measurements to support a PAT or and eventually a quality by design approach, and especially to develop assays that could be used to measure in-process viral titer. And what attracted us to LumaCyte and which brought us to evaluate the LumaCyte is a label-free method that they have available, which we then adapted to transaction monitoring. And also, we use this to measure transaction in the presence of gene of interest. And in addition to providing this method, a label-free method, LumaCyte also improved the accuracy and consistency along with providing an automated approach. And the measurement technology that is used by LumaCyte removes subjectivity and reduced error. Here's the experimental overview of the 3 different experiments that we did. Overall, we measured the real-time viral titer using the standard curve that we generated for the following systems. The first experiment was a baculovirus infection in SF9 cells, where suspension cells were seeded into 6-well plates and harvested 24 hours post-infection. Next experiment was the adenoviral infection in HEK293 cells, where cells were seeded into 24-well plates and harvested 48 hours post-infection. And the final experiment was the AAV transfection monitoring that we did in HEK293 cells, where we transfected using 3 different commercially available transfection reagents. And this was done in suspension cultures, which were then harvested 72 hours post-transfection for the Radiance measurements. And as Sean mentioned in a few slides earlier, for calculating a titer using the laser force cytology, the laser force cytology measures the biophysical or biochemical changes between cell populations with either infection, as we've seen with baculovirus or adenovirus, or treatment, as with the transient transfection or the transfection measurements, and also with changes in cell type or by use of a different subclone, for example. And in order to measure this, there are multiple metrics available, such as velocity and optical force index, which change upon infection or treatment, and they could be used to quantify cellular changes. One or more response metrics of the 20 that are measured could be identified with change proportionally and consistently with titer, and that could be used to quantify viral infectivity. And this is termed as the Radiance response metric. And multivariate models can also be utilized to correlate changes in the Radiance response metric to infectivity values. We then did the data analysis using the reporter, which was again discussed earlier by Sean. The reporter was used to expedite analysis and reduce time to results and generate fully automated reports. In this case, more than 20 unique parameters were trended. And for the baculovirus and adenovirus infection, a univariate metric was selected as the Radiance response metric, and this was correlated with the MOI. And for AAV transfection, there were 3 metrics that were selected to develop the Radiance response metric, and that correlated with transfection and also with the ddPCR results. And in addition to this, the reporter also gives scatter plots, velocity histograms, and calibration curves, which correlate the infection metric with MOI as shown below. So I'll go on to explain the first experiment we did, which is the baculovirus infection in SF9 cells. To summarize the experiment, we used suspension cells which were seeded into 6-well plates and infected at MOI values ranging from 5 down to 0.00032 in 5-fold dilution. And the Radiance analysis was conducted at different time points between 24 and 48 hours. And although the viral infection was detected at all the time points that were measured, the best correlation was developed using the 24-hour time point. And in the next slide, I'll show you the combined curve that we generated using the 24-hour data from 2 independent trials that were conducted over 2 separate weeks. So here's the 24-hour data for the SF9 baculovirus infection. Here, the two separate experiments showed excellent repeatability for the 24-hour point, and the Radiance response metric in this case was the average velocity. And in the highlighted region, we found a good trend from those four MOIs, and this was used to generate the calibration curve. Here's the calibration curve that we generated, which correlated the Radiance infection metric, the average velocity, with the MOI. And this demonstrates accurate and precise repeatability under similar growth conditions over two separate weeks, shown by trial 1 and trial 2. The table on the right displays the results of the cross-validation, and it demonstrates an accurate model prediction. The model is used to predict the MOI first, and then based on the predicted MOI, the TCID50 is then calculated using the equations below. And as you can see, the percent CV in this case compared to the stock titer of 2.03 E7 was 22%, but the measured value, the average measured value was 3.08 E7, and the locked-in difference was 0.127, showing the accurate and precise repeatability of this measurement. And as the next step, we would then use such a calibration curve to predict the unknowns in a real production setting or in a real process development setting. And that is shown here using LumaCyte's internal data, where the in-process samples of SF9 cells, which were infected at an MOI of 0.1, were tracked over a 6-day culture. And from day 1 to day 6, as you can see here in these histograms, the velocity distribution was tracked, and such information would then be used to generate a curve such as this, which predicts the viral titer, which is correlated to the Radiance infection metric. And so for an unknown sample where the Radiance infection metric would be measured, the viral titer could then be obtained. So Radiance allows infection to be tracked over time to ensure optimal harvest time point. And it also reduces the development time, enabling optimal processes, and could result in significant improvements in yield and process efficiency, and ultimately higher product quality. So to summarize the results of the first experiment with the baculovirus infection in SF9 cells, we were able to correlate the 24-hour post-infection, illustrating strong repeatability. The Radiance provided a rapid and accurate viral titration over a wide range of concentrations, as you've seen. And the in-process measurements could be made near real time once this calibration curve is generated, thus improving process optimization. All right, I'll move to the next experiment that we did with the adenoviral infection in HEK293 cells. In this experiment, the cells were seeded into 24-well plates and infected with MOI values ranging from 20 down to 0.027 in 3-fold dilutions. The Radiance analysis was conducted at different time points between 24 to 72 hours. And although the infection was detected at all time points that were measured, the best correlation was developed using the 48-hour time point. And again, in the next slide, I'll show you a combined curve within using these two independent trials conducted over 2 separate weeks at this 48 hours. And here's the data. And as you can see, there are similar trends at 48 hours across the 2 experiments. And here the infection metric in this case is the percentage of cells with velocity less than 2200 micrometer per second. And this, as you can see from the graph, it demonstrates excellent repeatability. And the highlighted MOIs were used to make the calibration curve that you see in this next slide, which here the correlation is between the Radiance infection metric, which in this case, again, is the velocity less than 2200 micrometers per second with the MOI. And the calibration curve shows repeatability between the weeks 1 and 2. And similar to the baculovirus case, the table on the right shows the results of the cross-validation, which demonstrates accurate model prediction. And also the model was accurate and precise over a larger dynamic range. And in this case, the percent CV that we observed was 12%, where comparing the average of the predicted TCID50, where we got 3.85 E8, and this is very comparable to the stock TCID50 at 3.56 E8. And the log-10 difference that we measured here is 0.034. And this type of this calibration curve could then be used to titrate for unknown samples, maybe using 2 or 3 dilutions and using this 48-hour curve. As Sean mentioned earlier, in addition to using this calibration curve, one could also use an absolute titer method to predict the titer. In this case, as you can see, at the 72-hour time point, we did see a plateau in the response at the levels of the virus added, and this was 100% normalized infected at these MOIs. And only using the cell count and the volume of the virus added without knowing the titer, one could predict the titer. And in this case, the predicted titer of 5.698 is comparable or within the range for the stock titer at 3.56 E8. Although it was slightly overestimated, I think having this method is a huge advantage when it is not possible to generate the calibration curve prior to using this for measurement. To summarize the results from this adenovirus infection experiment, we did get an excellent fit at the 48-hour post-infection with strong repeatability. And the results that we collected were consistent with the previous Radiance measurements and thus demonstrating strong reproducibility. The last experiment I'll be discussing is the experiment we did to measure the AAV transfection in HEK293 cells without labeling. So HEK293 cells in suspension were transfected with 3 different reagents which are commercially available. And on day 3, samples were taken to or harvested to measure on the Radiance, although the culture carried on. And at the end of the culture, the ddPCR measurements were also performed. In this case, there was no use of labels or antibodies. And such a measurement would be a huge advantage, especially in the case where a gene of interest is used during development. And one could then obtain the transfection efficiency during the course of the culture and could greatly aid in terms of not only selection of the transfection reagent, but also in terms of titrating for the amount of transfection reagent to add to the culture. And in this particular case, the ddPCR titers also correlated very well with the same Radiance metrics, although in other cases, it is possible to still correlate the ddPCR titers with the Radiance response metrics and use that to predict ddPCR titers ahead of the titer measurement. Here are the results of the AAV transfection reagent testing. For the 3 different transfection reagents shown as TR1, 2, and 3. The transfected population showed an increase in optical force and also lower velocity compared to the cells which were not transfected, which is shown in the gray. And the percentage of cells with velocity less than 2400 micrometer per second, along with the standard deviation of velocity, was used as the Radiance response metric in this case. And when comparing the transfection reagents 1, 2, and 3, the dotted line, which show the percentage of cells with the velocity less than 2400 micrometer per second, these show the transfected cell population. And the transfection reagent 3 showed the highest transfected population, followed by the reagent 1, and that was followed by reagent 2. And these results were also confirmed from the ddPCR measurements that we did outside of this experiment. So thus developing a correlation between the Radiance metrics and the ddPCR results allows the users to use Radiance as an early predictor of ddPCR, leading to an in-process real-time AAV process development assay that we currently do not have. And this could enable rapid monitoring of AAV production and process condition optimization. And here is the result showing the Radiance response metric with the various transfection reagents that were tested and the control, which was not transfected. And as you can see, the transfection reagent 3 had the highest Radiance response metric, followed by 1, and that followed by the transfection reagent 2. And on the right side, you see the ddPCR titer correlated with the Radiance response metric. Using such measurements, it is possible to reduce the time to select the optimal transfection reagent. And in this case, that was a result for us where we were able to select the transfection reagent based on the Radiance response metric data. And the results were obtained near real time with about 5 minutes analysis per replicate. And the laser flow cytology quantitative measurement has potential to significantly improve process development timelines and eventually impact quality and the production yields. So to summarize this experiment for the HEK293 transfection with 3 different transfection reagents, we saw a strong correlation between Radiance metrics and transfection, and Radiance could be used to develop a predictive correlation for ddPCR. And the data suggests that Radiance could be an early predictor, not only for transfection, but also for AAV titer during process development. So overall, to conclude from these 3 experiments, we saw marked improvements in speed and accuracy using the Radiance label-free LFT assay. So for example, for the SF9 baculovirus titer determination, which is currently done using a plaque assay and takes 1 week, can now be done 24 hours post-infection. The viral titering at 5 in HEK293, which is currently done using TCID50 and takes 3 to 6 days, can now be done 48 hours post-infection. And measuring of AAV transfection real time, even in the presence of GOI, could be currently done, which was previously not possible. And thus, it provides a near real-time PAT analysis tool. And also, the Radiance could be an early indicator of titer in these HEK293 cells. And this is, again, currently not possible. So this would give a new PAT tool to predict titer in these transient transfection experiments. So overall, we could get a significant time and resource savings, and it could accelerate our current assay workflows and provide a cost savings across time, reagents, and resources, and reduce the errors through improved accuracy. And overall, we realized several improvements, including rapid analysis, enabling in-process measurements, reduced sample load, reduced labor, and could eventually support manufacturing runs. So with that, I'm going to conclude my part of the presentation, and we are open to questions. Thank you for your attention.
Okay, thank you, Bhargavi. Let's move on now to our Q&A. And there's still time to submit your questions using the Q&A tab on the left side of your screen. We have lots of great questions already. We will try to get to as many as possible. Our first one here comes in for Sean. They want to know, they're asking, it takes our analytical team 1 full week to provide us our TCID50 results. If our group adopted Radiance, how long would it take to receive these same infectivity results, and why is it so much more sensitive?
Sure, great question. So in general, Radiance is between 3 and 10 times faster than a traditional infectivity assay such as plaque or TCID50. It really just depends on the viral system and the cell line that you're using. So there is a range. So typically a 7-day assay would be 1 or 2 days post-infection using Radiance versus the 7 days. I think the second part was how or why is it so much more sensitive? So Radiance is measuring phenotypic changes in cells based upon those intrinsic biochemical and biophysical changes. And so one example that I shared earlier was when virons are produced inside a cell, it changes the refractive index, which we're very sensitively able to detect, by integrating forces across the entire cell, which leads to the change in velocity, which is one of the principal, measurements of optical force that we make. So that's how we're able to do that. And I think the last example Bhargavi gave with AAV, right? That is a system that doesn't produce cytopathic effects. So, being able to detect that or lentivirus also highlights the ability of the system or the sensitivity of the system in detecting such viruses.
And thank you, Sean. They also had a follow-up noting that their viral system takes approximately 8 to 10 days to produce plaques. So how is Radiance able to detect infectivity sooner than traditional assays?
Sure. Well, so for the plaque assay, as everyone's aware, that's sort of a final endpoint, right? Because the plaque is the clearing of the cell. So not only have they been infected, but they've run their course and disintegrated, so leaving that clear area on the monolayer. And so we're detecting, subtle changes in cells, subtle morphologic, right, biophysical, and then the biochemical differences, as I mentioned, that gives us the ability to really detect much sooner.
All right. Bhargavi, what are the advantages of implementing Radiance as part of the AAV process development workflow?
Sure, yeah, that's a great question. Thank you. Radiance provides an early read on key measurements that are currently not possible, as I've shown in my presentation. For example, the effect of process parameters, especially the different DNA vectors, DNA vector ratios, as well as transfection reagents, and all of these on transfection efficiency could be measured, and that's allowing for improved process design and could lead to higher yields and an efficient process. In addition to the ddPCR titer and any other in-process attributes, upstream could have a near real-time PAT tool for measuring infectivity prior to process selection and lock. So I think that's a huge advantage that currently doesn't exist.
All right, thanks for that. Sean, what is the footprint of the device and any other installation requirements?
Oh, sure. So it just requires a standard laboratory bench, right? A stable work surface to install it on. It's roughly, you need 2 by 2, roughly by 3 feet in height. It uses standard power, and the only other requirement is house air of about 80 psi. So that's it. It's pretty minimal requirements.
Great. So Bhargavi, with subjectivity and errors being the norm with traditional infectivity assays, what type of error rate do you typically see with Radiance?
Sure, that's a good question. So while Radiance could still be subjected to the technical pipetting errors similar to TCID50 and plaque assays, the subjectivity due to qualitative measurements such as counting plaques or estimating the percentage of cells that show the cytopathic effect, that is replaced by precise optical force measurements with Radiance. So the instrumental errors have been reduced to minimum and far below the typical biological changes. So typically with Radiance, you could expect to achieve 0.1 to 0.2 log10 error or better, that I've shown already in my presentation. And this compared to a 0.3 log10 error or higher with traditional assays is a huge improvement.
All right, thank you. Sean, in what ways does Radiance reduce error compared to the traditional assays, plaque and TCID50?
Sure. So Radiance is a fully automated quantitative measurement to replace plaque or TCID50, and it removes subjectivity user to user. In addition to the painstaking efforts that we have taken in developing the instrument to reduce the error and to make the measurement as repeatable as possible, as I showed with a percent error of less than 1 over almost 20 days. In addition, fewer dilutions are required, right? Because the instrument dynamic range is typically 3 to 5 logs with viral concentration. So more replicates can be run versus dilutions to get a reading, which obviously increases your accuracy there as well.
All right. Bhargavi, can you measure viral titer from adherent cells with the Radiance system?
Sure. Yes, we can. And although Radiance is a flow-based system, any enzymatic-based detachment reagent will work well to detach the cells. For example, triple treatment followed by the Radiance measurement has been shown to work very well.
Okay. Sean, is method development difficult with Radiance? And how easy is the instrument to use?
I'll start with the last part first. So the instrument is very easy to use. It's designed to be, click, play, and walk away. We've had students learn to use it in just a few days and have been, conversant in it and able to run it and get data off and such. In terms of method development, this is also easy, right? Principally because we're so much faster, right? If you're not waiting a week to get your results, right, you can iterate on the method development, much more, and have a much faster turnaround time, right? Being one of the key advantages. For example, we had a demo with a customer a number of years ago where they were looking at 4 systems, 3 of which were completely new to us, right? We had no background. The other was adenovirus, which we have a lot of experience in, but they were able to do that, those 3 unknowns and adenovirus in the first week and then reproduce them in the second week. So that just gives you a feel. If you can get through 4 systems in 1 week and then reproduce it well in the second week, that's not a very difficult system, right? It makes it much easier to get through the method development than a traditional assay, and that's one of the big advantages we feel.
All right. Bhargavi, are you also planning to look at real-time AAV infectivity measurements?
Yes, I mean, that would be the next main area that we would like to use this Radiance for once the method becomes available. So currently a method is being developed at LumaCyte that we're looking to evaluate. LumaCyte is currently working to overcome the current limitations of the AAV infectivity measurements by using their nuclei analysis as opposed to the whole cell analysis method, and this could increase the sensitivity besides the current method of co-infecting with adenovirus. So yes, whenever this becomes available, I think this would greatly enhance process development, helping us select for the process where we also measure the AAV infectivity in addition to the typical measurements we do, such as ddPCR titer.
Okay, thank you. And this next question will be our final question. We had a lot of great questions today, and we couldn't get to all of them, but we'll try our best to get back to everyone who submitted personally after this webinar. And Bhargavi, can Radiance be used for cell subcloning or cell line development?
Yes, that's a great question. Although we haven't used it for that application, I believe so that we could use Radiance for identifying the differences, especially the phenotypic differences between different cell types and cell clones, and then correlate that to the yield from those subclones. And Sean, do you want to add to this?
Yeah, sure, absolutely. So that's a really great application, right? Cell banking and cell line development. It's something that that we've worked with customers on before, and it's a totally great application for applying Radiance. In terms of cell banking, being able to monitor changes within a cell line, changes over passage from time, is really critical for maintaining quality. With the cell bank or cell line. So that's a great application. Yeah, it's a very good question. Thank you.
Thank you. And thanks to everyone listening for attending this Fierce Biotech webinar and submitting so many great questions. I'd like to thank our speakers for participating and Catalent and LumaCyte for presenting today's webinar. This webinar has been recorded. You'll be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining us. We look forward to seeing you at future events.